Morning students. Today we will learn about the chapter 6 life process from class 10. Let's understand what is life processes. Life processes are the important or vital processes carried out by all the living organisms in order to maintain and sustain the life. We know that living organisms are made up of cells. Cells combine to form the organ, and many organs together form the organ system, making the organism. Whether it is a cell, tissue, or organ, all these are made up of molecules. And when these are interacting with the environment, there is a chance of these to break down. So, to keep repairing and maintaining these structures which are made up of molecules must have movement of molecules. Also, molecular movement is essential to carry out the life processes. The main life processes include nutrition, transportation, metabolism, respiration, reproduction, and excretion. In this chapter, we will be understanding better about all these life processes. Maintenance of the different processes that are needed for the life need energy. And where do we get energy from? We get energy for all the process from outside the body. So how do we take this energy into the body? The process that is commonly called nutrition is used for acquiring this energy. And thus, nutrition is the process of being able to intake food and utilize it for energy purpose. Transportation. There are different kinds of molecules which are moving within the body. We have blood which moves to different parts of the body, and blood carries a lot of nutrients. So, we need transportation to be happening within the living beings. Metabolism. Our body is a storehouse for many chemical reactions, both the making kind of reaction, that is the reactions which make molecules, and the reactions which break molecules. So this is metabolism. We generally intake oxygen and give out carbon dioxide. In the process, we oxidize certain nutrient molecules, mostly the carbon molecules, to give out energy and carbon dioxide. This is respiration. Then we have reproduction, that is giving birth to the young ones. And then due to metabolism, due to respiration that is occurring within the body, there are some waste products which are synthesized in the body, and these have to be excreted out. This process is called as excretion. Nutrition. The process by which living organisms procure food and utilize it for obtaining energy and for building body is called as nutrition. Life on Earth depends on carbon-based molecules, and the food source, most of the food is made up of carbon. Let's now understand the different types of nutrition. We have autotrophic nutrition and heterotrophic nutrition. Autotrophic means self-made. Hetero means different. So these heterotrophs depend on other plants or animals for their nutrition. So autotrophic nutrition is a mode of nutrition in which organisms prepare their own food by the process of photosynthesis. Here, Simple inorganic substances like carbon dioxide and water in the presence of sunlight and chlorophyll are used to synthesize organic food. So green plants are autotrophs. Heterotrophic nutrition is the mode of nutrition in which organisms obtain their food from other plants or animals. Fungi, bacteria, and animals are the heterotrophs. Types of heterotrophic nutrition. We have saprophytic, parasitic, and holozoic. In saprophytic, the organisms get the food from dead and decaying organisms. This is a chemoheterotrophic extracellular digestion. That is, digestion is happening outside the cell. Here, they break down the food material outside their body and then absorb it. The second method is the parasitic mode of nutrition. Here, the organism derives nutrition from plants and animals without killing them. For example, we have cuscuta, which is a plant which grows on bigger plants and derives nutrition from them without killing them. We have ticks, which we can see in hairy animals. Lice you can see in human hair also. Leeches are ectoparasites and tapeworm are endoparasites. Ecto means outside the body of the organism and endo means the parasites which are within the body of organism. The third method is holozoic method. Here, the organism takes in the whole material and digests it within the body to derive nutrition. So here we have examples like humans, birds, amoeba, and paramecium also. Let's now see how amoeba and paramecium obtain their nutrition. Amoeba makes these finger-like structures which are called as pseudopodia. These structures which are called as pseudopodia. These pseudopodia encircle around the food particle and the food particle digests within this food vacuole and the food is broken down into simpler substance which is absorbed by the cytoplasm and the waste is then thrown out again through the cell surface. But in case of paramecium have definite shape and the cilia, these thread-like structures which are there all over the body, which are called as cilia, help in pushing the food particle through to this oral groove. Once the food enters into the body, the food is digested inside. It is broken down into simpler substances. The undigested food and the waste particles are then thrown out through this anal pore. So here we can say that when the food particle is taken in, the process of ingestion is happening. Ingestion means taking in of the food. And here, after the food is encircled by the pseudopodia, digestion starts occurring. That is, breaking down of the complex molecules into simpler molecules starts happening. And after this, the simpler molecules are absorbed and they are sent into the cytoplasm to all parts of all the spaces in the amoeba. And then at the end, the undigested food is thrown out. So that process is called as digestion. So in amoeba, we can see ingestion, digestion, absorption, assimilation, and egestion. In human beings, digestion and absorption of food takes place in the digestive system. The digestive system consists of an alimentary canal and the associated glands. The alimentary canal, in case of humans, is basically a long tube which extends from the mouth to the anus. This tube is further differentiated into different parts, and these different parts are specialized to perform different functions. Now here in this diagram, we are seeing the human alimentary canal, which we have been studying even in the lower classes. So the alimentary canal starts from the mouth and then it extends to the anus. Starts from the mouth and it extends to the anus. So after mouth you have the esophagus. So this pipe is the esophagus, which extends into a small bag-like structure which is called the stomach. Stomach then extends to form this coiled pipe over here, which is called as small intestine. So before the stomach becomes the small intestine further, you can see here one more organ which is called as the liver. Liver has the small structure which is called as bile duct. Also, there is another structure over here which is called as pancreas. Both the liver and the pancreas give out its secretion into the duodenal area, which is a part of the intestine, and the food gets mixed up with that and then enters into the small intestine. So the small intestine is about 6 meters long in case of human beings. So the small intestine will further open up into the large intestine. Here it forms the large intestine and then opens up as anus. Here we'll observe a few more parts which would help us in understanding digestion in human better. Here in this first diagram, we see the peristaltic movement in the esophagus which will push the food down to the stomach. In the second figure, we see the pharynx. Pharynx is the body cavity that connects the nasal and oral cavities with the larynx and the esophagus. Here you have the sphincter. So sphincter muscles are muscles which will constrict and relax. This constriction and relaxation will help in either stopping when it is constricted, and when it is relaxed, it will allow the food to pass through. Similarly, you have another sphincter muscle over here which is called as pyloric sphincter. This is called as pyloric sphincter. Okay, the last figure that you see here is that of the section of intestine. So in section of intestine, I said there are villi, that is the finger-like structures which help in absorption. So you can see that the villi inside have a lot of capillaries. These capillaries will help in absorption of the nutrition, and from this the nutrients that are absorbed are sent to different parts of the body. Let's understand the term biocatalyst. Bio, life, catalyst is any substance which will increase the rate of reaction. So biocatalysts in case of living organisms are the enzymes which help to break down the complex food particles into absorbable smaller particles. So we have examples like salivary amylase, pepsin, lipase and trypsin. Let us now understand the process of digestion. The food first enters the mouth. The mouth has teeth which crush the food and make it into smaller pieces and of the same texture. Mouth also has salivary glands which secretes saliva. That is, the saliva has salivary amylase or ptyalin. This enzyme breaks down the starch in the food to simple sugar called as maltose. The food then enters into pharynx, which is a common opening for the respiratory system and the digestive system. From here, the food enters the esophagus, that is the food pipe. Esophagus shows the peristaltic movement, that is the rhythmic contractions you can see in esophagus, which will help the food to pass through the pipe and then enter into the stomach. Stomach has gastric glands which secrete hydrochloric acid, pepsin, and mucus. Hydrochloric acid activates pepsin and kills the bacteria. If the acid is not present, then the pepsin does not become an active form. Pepsin, it breaks down the proteins to simple absorbable compounds called as amino acids. And the mucus protects the inner lining of the stomach from the action of hydrochloric acid. The food from stomach then enters into the small intestine. Before it enters into the small intestine, the food is mixed by the bile juice which is secreted by the liver. Bile juice makes the food alkaline for the pancreatic enzymes to act. Otherwise, if the food is acidic, the pancreatic enzyme does not act on the food. Also, the bile salts break down large fat globules into smaller globules. That is, emulsification of fats occurs because of bile juice. And also the food is mixed with pancreatic juice which is released by the pancreas. The pancreatic juice consists of trypsin which breaks down proteins, and it also has lipase which breaks down the lipids. So the small intestine, the food that enters the small intestine is mixed with the hydrochloric acid, pepsin, mucus, bile juice and pancreas, and when it enters into the small intestine, the food is then mixed with the intestinal juice which helps in complete absorption of the food by breaking down the already partially digested food into completely digested food. Here, complete digestion takes place. That is, proteins are finally broken down into absorbable compounds called as amino acids, carbohydrates are broken down into glucose, and fats are broken down into fatty acids and glycerol. The food then enters into the large intestine. Large intestine has villi which absorb water. So in the small intestine, we have villi which I have already shown you previously, and the villi has lot of blood vessels, blood capillaries, which absorb the digested food. And from the blood vessels, that is the capillaries, the nutrients go to different parts of the body for building the body or repair work. And the rest of the food that is not digested then enters the large intestine. So large intestine has again villi which absorb the water which is there in the food, and then the undigested food is thrown out through the anus. Thus, the process of digestion can be divided into the following steps. 1. Ingestion, that is intake of food. 2. Digestion, that is breaking down the complex molecules, the complex food particles into simpler molecules. Absorption, that is absorption of the simpler molecules into the bloodstream. Assimilation, assimilation is to spread out the nutrients from the villi to different parts of the body. Then egestion, that is the excretion part, that is excreting out or eliminating the undigested food from the body. Okay. So the energy that is derived from the process of digestion, it can be sent to different parts of the body, or it can also be stored. So this stored form of energy in case of animals is glycogen. Glycogen is generally stored in muscles and liver. The length of a small intestine in herbivores is longer than carnivores. The cellulose takes longer time to digest. Hence, the herbivores need a long intestine to allow the cellulose to digest. A point to note, the herbivores themselves cannot digest the cellulose. They harbor anaerobic bacteria in their intestine which helps in breaking down this cellulose. On the other hand, carnivores have shorter intestine because meat is easier to digest. Now, we will learn about nutrition in plants. Green plants are autotrophic. Auto means self. Trophic means nourishing. So they nourish themselves. Green plants synthesize their own food by the process of photosynthesis. Green plants use carbon dioxide and water to make carbohydrate, that is the food that is needed by them for energy and for building the plant body. So this carbon dioxide and water is taken up in the presence of sunlight and chlorophyll to make carbohydrate. Carbohydrates are stored as starch, and this is the internal source of energy for the plant. So starch is nothing but a complex carbohydrate. So here we are seeing the equation of photosynthesis. Here we see 6 molecules of carbon dioxide have combined with 12 molecules of water in the presence of sunlight and chlorophyll to give rise to glucose. 1 molecule of glucose, 6 molecules of water, and 6 molecules of oxygen is released out. Let us understand the different events that occur during photosynthesis. So the different events are: 1. Absorption of light energy by the chlorophyll. So during day, the chlorophyll that is present in the leaves absorb light from the sun. And this light energy is converted into chemical energy by splitting of water molecules into hydrogen and oxygen. And the last step is the reduction of carbon dioxide by hydrogen to form carbohydrate. So the water molecules that are split into hydrogen and oxygen, the hydrogen that is formed by the water molecules, splitting of water molecules, is used up by carbon dioxide to form carbohydrate. Now this sequence may not be same in some plants like that of the desert plants. In desert plants, carbon dioxide is taken up at night. Then they prepare an intermediate compound. This compound is then acted upon by energy which is absorbed by chloroplast during the day. So the intermediate compound is formed at night, and this is then converted into the carbohydrate, which is starch. Here we are seeing a cross section of leaf. So the uppermost layer is the cuticle. So here this layer is the cuticle. Below the cuticle layer, you have a layer of epidermal cells, which is called as the upper epidermis. Below that, you have the palisade parenchyma. These are the palisade. I have another diagram I will be showing you. This is the palisade parenchyma. And below this, you have the spongy parenchyma. And in between these parenchyma, you can see the air spaces which are there. So which open out through the stomata. The stomata are in turn guarded by the guard cells. These cells close and open for the stomata to allow or not allow the air to enter and exit from the lower epidermis. So the last layer is the lower epidermis. So here is a 3D diagram. Here we can see the upper epidermis. So above the upper epidermis, here you have the cuticle, which we saw in our previous picture. So below the upper epidermis is the palisade parenchyma, and below the palisade parenchyma, you have the spongy parenchyma. And below this, you have the lower epidermis. So here they say it as mesophyll cells. That means the whole of this layer is called as mesophyll. Therefore, they call it as palisade mesophyll cell. And here they call it as spongy mesophyll cells. So in between, you have the veins. And here you can see the stomata, which is guarded by the guard cells. So here is a 3D diagram. Here we can see the upper epidermis. So above the upper epidermis, here you have the cuticle, which we saw in our previous picture. So below the upper epidermis is the palisade parenchyma, and below the palisade parenchyma, you have the spongy parenchyma. And below this, you have the lower epidermis. So here they say it as mesophyll cells. That means the whole of this layer is called as mesophyll. Therefore, they call it as palisade mesophyll cell. And here they call it as spongy mesophyll cells. So in between, you have the veins. And here you can see the stomata, which is guarded by the guard cells. Let us now learn more about the raw materials that are needed for photosynthesis and the essentiality of these. First, carbon dioxide. Carbon dioxide enters the plants through the tiny pores called stomata on the surface of leaves. The opening and closing of the stomatal pore is controlled by the guard cells. The guard cells swell when water flows into them, causing the stomata to open, and when the water flows out, the guard cells will shrink and the stomatal pore closes. An enormous amount of gaseous exchange takes place through the stomatal pore during photosynthesis. So carbon dioxide enters and oxygen exits. Exchange of gases not only occurs through stomatal pores but also occurs in stems and roots. Let us now understand the importance of carbon dioxide by this experiment, which is given in your NCERT textbook, activity 6.2. Here you have two bell jars. If we keep these two bell jars in a dark room for 3 days, and then now we place each plant on a separate glass place, and we put potassium hydroxide in one of these bell jars. So here we have the potassium hydroxide, but in the other place we do not keep the potassium hydroxide. And we seal these bell jars with Vaseline so that no air enters them. So the plant has only the air which is there within this bell jar. Now we know that potassium hydroxide will absorb all the carbon dioxide from the air. So in A, since potassium hydroxide is there, it absorbs all the carbon dioxide. Whereas B, we do not have potassium hydroxide, so carbon dioxide is still present in the air surrounding the plant B. Now if we do a starch test for both of these plants, we see that the presence of starch is greater in case of B because of presence of carbon dioxide than that of A, which suggests that carbon dioxide is important for photosynthesis. So this is your textbook diagram where we see the guard cells. You can see here guard cells. Here the stomatal pore is open and here stomatal pore is closed. Chlorophyll pigment is present in chloroplast. Chlorophyll traps the light energy which is essential for photosynthesis. Now this we can see with the help of experiment where a potted plant with variegated leaves. Variegated leaves is the leaf which has different colors or irregular patches of different colors. For example, you have the money plant or the crotons. Now keep this plant in dark room for 3 days. Why do we have to keep in the dark room? So that whatever starch is already present in the plant is used up by the plant for whatever life process are occurring within the plant. Now, after 3 days, we keep this plant in sunlight for 6 hours. Now, if you pluck a leaf from the plant and mark the green areas in it and trace it on a sheet of paper, and if we take this leaf and then boil in water for a few minutes. Why do we boil? We boil so that we kill the cells in the leaf. That is, the leaf is cells in the leaf is killed. And then we dip the leaf in the alcohol. Alcohol will remove the chlorophyll and it gets depigmented. Now we can test these leaves for iodine. So if we add iodine solution, the place that has starch will turn blue-black, whereas the place that does not have starch will not take up the blue-black color. This suggests that chlorophyll is important for photosynthesis. Water that is needed for photosynthesis is absorbed from soil by the root through osmosis. Other than this, the plant also needs some other elements like nitrogen. Nitrogen is an essential element and is used to synthesize proteins and is also a part of other compounds like the DNA and RNA. Other materials like phosphorus, iron and magnesium are taken up from the soil. Thank you. If this has been helpful to you, please give a like. And for rest of the chapter, please continue to watch this channel. Thank you.